Anyway, good morning, and I appreciate you for your midterm one exam. I went through the your answer script, and you did very well. This is really a good sign during this COVID-19. That means you are on track and you are studying your subject very seriously. Today, we shall discuss on another topic, very important, uh, secondary metabolite, a class of compound which has been used by people in ancient time. And those metabolites have huge influence in human civilization as well as in agriculture, medicine. They are alkaloids. So I shall, among the secondary metabolites, alkaloids have been used by human since ancient time. Before I start the lecture, I also would like to discuss a little bit about your assignment, not exactly assignment, a comprehensive review on pheromones in agriculture, pheromones in biorational management of crop plants or like that. So yesterday or day before yesterday, I had a talk with Joy Debnath. I asked him to coordinate the job. So what you need to do, you need to cover all the literatures available on pheromones in agriculture. This is why it is better for you to make a content of the review, then collect all the literatures by browsing Google Scholar, uh, PubMed, Science Direct, and other scientific databases. After having all the data, now it would be the ideal, you can divide the data in different groups depending on the sections of your review. So your review obviously have a title, an abstract, an introduction, uh, and then diversity in pheromones or like that, chemical diversity of pheromones. Uh, Next, you can go for chemical diversity means different class of secondary metabolites and different class of pheromones also. Diversity of pheromones would be classes of pheromones. Second, you will go the chemical diversity of pheromones. That is, some of them are alkaloids, some of them are terpenoids, some of them are phenolic or other compounds. And next, you will go the role of pheromone in insect biology. So you can make a list which pheromone is doing what kind of function in the insect. And next section would be application of insect pheromone in the biorational control of insects. In that case, different pheromones people have been using in the practical field as well as in the laboratory conditions. Some of the new pheromones have been tested. So what you do, the name of the pheromone, one column. Second column is the source of pheromone. That means which insect is producing, and then the application of pheromone in agriculture, for example, control of male insects or female insects, specific insects, or like that. And then mode of action, if you know, for example, sex pheromone or other thing. Last column should be the reference. Reference, for example, Korim et al. 2010. You can make a good table. And finally, you can write another section, prospects and challenges of pheromone application in agriculture, because there are high prospect, and every country are now using pheromones, but there are some limitations also. So you can put the limitations as well as the prospect. Many pheromones are still unknown, and some of them are in a trace amount, and difficult to trap, a good amount to detect. So many of the insect pheromones are out of the detection. So it is a big challenge. Structure determination of small amount is really a big challenge. Other challenge is that pheromone, some of the compounds are stereochemically active and small molecule, but very difficult to synthesize. If you cannot synthesize large amount, then it is a problem. Uh, the limitation, if you see, one limitation is pheromones. Some of them are very a powerful volatile compounds. So after application in the pheromone trap, within a few hours, they are evaporated, and then you have to apply it again or like that. And you have to make formulations so that it is slow released or like that. Pheromone use is specific for specific insect. It is a big advantage, but it is also limitation, because farmers sometimes feel that after application of this pheromone, all the insects could be controlled, and this is why when they see other insects are damaging their crop, they become frustrated. So it is important to train the farmers and tell them you are controlling a particular insect by using the pheromone trap. Uh, and you can gradually go to the conclusion and a list of reference. I believe you four students would be four authors, and it would be a nice review. And this review will be considered your assignment, and I shall put 10 marks on it. And I will also consider it for publication. Hopefully, it will be published in 2021 if you can make a good report. And during preparation of your review, you must collect all the chemical structures, because structures, one figure would be the structures of pheromone used in agriculture. That means the compounds that have already been tested in agriculture or management of agricultural insects. Uh, uh, those structures need to be drawn uh, by yourself, and then to add as a figure. Uh, in our laboratory, Mahmoud has CamDraw software, and it is very easy to draw any structure. So what you need to do whenever you read the paper, by using a snipping tool, or somehow make a copy of that structure, uh, then you can collect all the structures and name, and finally you can draw by using the CamDraw. I guess Rahat is now in the university. He can easily do this job by the help of Mahmoud, because Mahmoud would offer you the CamDraw software, and it may take time, but it would be good experience, because it would be a good skill also. Rahat, is it okay?
Yeah. Yes, sir. I can do that.
Okay, fine. So Rahat has one job, and Joy Devnath, what you need to do, you make an outline and share with the members uh, what would be the content, and then ask them which part would be, addressed by, sorry, by which student. and then start the job.
Yes, sir.
So
Okay, sir.
what you need to do, you need to fix some of the keywords. For example, insect pheromone, sex pheromone, or pheromone trap, pheromone in agriculture, like that. Uh, and using those keyword, you can browse the literature. And what you need to do, maybe if you do the same job by code and get the same paper, no meaning. In that case, you can target, for example, 2015 to 2021, someone, and then 2010 to 2015, someone. Like this, you can separate the timeline of literature, and in the uh, browser, you can also set the timeline, um, and get the literature from there. And after having all the literature in a folder, then you can share. And good idea to have a Dropbox folder that you can share with you, among you as well as with me. So, do you have any question on this topic? And is it okay with you?
Yes, sir. I already started the job. I downloaded several papers about 2025 and organizing them as in different folders in my PC, as you have told earlier, sir, based on their keywords. And now, as you have given me outline, I will ready it and share it to others. As
Excellent.
well as I also create a Dropbox folder and share it into the group, sir,
Excellent. And
so that everyone can.
another point is that you need to check at least two journals. One is Journal of Chemical Ecology. So if you go to the Journal of Chemical Ecology, within this journal, you can easily browse. There you can find some browsing system. So, Journal of Chemical Ecology, it is a specialized journal for this type of topic. Another journal is Chemoecology. So you can also uh, search new literature on published in chemoecology. You can find other sources, for example, American Chemical Society. They have lots of journal on natural products, uh, and Royal Society of Chemistry. So visit their website uh, and browse their journals also, in addition to Elsevier, Springer, and many other journals. But the most important point is you may find some review also. So the review, if you find, then that would help you to get lots of literature there. And review, when you find a review, you can see many people cited the review, and check all the cited papers, and you can find some good papers over there also. So there are lots of ways to cover all the major literatures published in this topic. And getting all the literature is very important. And if you go through the past reviews, then you can organize the content of your the review. Already I told you a skeleton of structure of your review, but you can polish it further. Now, Fatima, do you have any comment, or uh, Rahat and Shafi? Shafi is doing another job, because in BTL 611, he is only the person, a student, because um, Ruhul is left. So he is writing another review on seed endophytes. This is why please give him a less load, obviously one section, but less load to him. Uh, Kanis Fatima, can you hear me?
Yes, sir. Yes,
Okay.
sir.
Uh, do you have any comment? Is it agreeable with you?
Yes, sir. It's agreeable to me, and I also doing some job like Joy Devnath downloaded some papers. I also downloaded some papers, and we're thinking about a meeting today or tomorrow about this.
That would be great to have a meeting. And one important point is that deadline is February 7. So by February 7, you have to complete your review, so that you have to make a precise plan so that you can do it by February 5th, and then share among yourself and combine all the small parts and make the whole review and polish it, and then submit to me. Rahat,
Yes, sir. We will try.
Yes, sir.
is this
We,
plan make sense?
yes, sir. The plan is good. Now we must get to start the
Okay.
work.
Shafi?
Yes, sir. It's okay for me.
Okay, very good. Then we shall discuss a little bit on uh, alkaloids today. Uh, those who attended my lecture on bioactive natural products, they might be familiar to alkaloid. Alkaloid, you can see this image. This image actually is very important internationally as well as in the alkaloid, because uh, the first individual alkaloid, morphine, was isolated in 1804 from a poppy. This is a poppy flower, and this is the pod of poppy. So what the alkaloid producer do? They scratch on the pod, and milky substances are coming out, and those milky substances is a very complex secondary metabolite. You can see the structure here, and this structure was this compound was isolated 1804 uh, by a German scientist, possibly from a Papaver somniferum, and named it morphine. And before, purification of this or isolation of this compound, people have been human beings have been using morphine since ancient time as a drug, elusive drug, as well as a painkiller. And Morpheus, the morphine, the word comes or came from the Morpheus, a Greek word, means Greek god of dreams. So alkaloids are a group of naturally occurring chemical compounds which mostly contain basic nitrogen atoms in their structure. And already I defined when I discussed on secondary metabolites. So alkaloids, in addition to carbon, hydrogen, and nitrogen, alkaloids may also contain oxygen, sulfur, more rarely other elements such as chlorine, bromine, and phosphorus. That means alkaloids has a huge elemental diversity in their structure. Even phosphorus, bromine, chlorine can be present there. This is why biosynthesis of alkaloid is very expensive for the plant, because it requires lots of different uh, elements like nitrogen. Nitrogen is always a limiting factor in the environment for the plant, and it requires for growth and development hugely. Uh, but phenolic compound or terpenoidal compound, they require carbon, hydrogen, oxygen, and plant has lots of carbon, hydrogen, oxygen, as they have the photosynthetic mechanism. Alkaloids are produced by a large variety of organisms, including bacteria, fungi, plants, animals, and are part of the group of natural products. Um, Different diverse group of organisms can produce alkaloid. Many alkaloids are toxic to other organisms, and they often have pharmacological effects and are used as medications, as recreational drugs, or in entheogenic rituals. Generally, alkaloids are in beta test. Alkaloids, uh, most of them are used by the producer as a defense compound. So origin of the word alkaloid, I already mentioned the origin of one compound morphine. The name alkaloids, it came from a German word alkaloid, was introduced in 1819 by a German chemist, Karl Meissner, a very famous chemist, and is derived from late Latin root, Latin word alkali. That means alkaloidic comes from the Latin word alkali, which in turn comes from Arabic word alqualja, ashes of plants. That means the original root of alkaloid is alqualja. Alqualja, alkali, alkaloidy, and then alkaloid. So it was evolved this word in a long path. And the meaning of alkaloid, ashes of plants. And the suffix Greek means like. However, the term came into wide use only after the publication of a review article by O. Jacobson in the Chemical Dictionary of Albert Leidenburg in the 1880s. German since early uh, 17th or 18th and 19th century, German chemists dominated worldwide. If you see, just go 30 years before, almost all the chemistry Nobel Prize owned by the German chemist. More than 12,000 alkaloids had been identified by 2018. Actually, latest data I have no, but if you find in the literature, please share me. Maybe now 15,000 or like that. There is no unique method of naming alkaloids. Many alkaloids names are formed by adding the suffix "ine" to the species or generic alkaloids. For example, atropine, ine, atropine, uh, discovered from Atropa belladonna. Atropa belladonna is a plant. Strychnine is obtained from the seed of strychnine tree. If several alkaloids are extracted from one plant, then their names often contain suffixes like iodine or anine, alene, or inine, etc. There are also at least 86 alkaloids containing the root vin extracted from the vinca plant. For example, vincristin or vinblastin, very famous, anti-cancer drug. So alkaloids containing plants are used by humans since ancient times for therapeutic and recreational purposes. For example, Mesopotamia, at least around 2000 years before the birth of Christ. Here you can see an image. The great poet Homer, he was blind. The Odyssey of Homer, very famous, book, Odyssey, the you know uh, book of poems by Homer, referred to a gift given to Helen by the Egyptian queen, a drug bringing oblivion. It is believed that the gift was an opium-containing drug. A Chinese book on house plants written in first to third centuries before the birth of Christ mentioned a medical use of ephedra and opium poppies. Also, coca leaves were used by South American Indians since ancient times. It is now clear that alkaloids have been used since ancient time for therapeutic and recreational purposes. Uh, German chemist Friedrich Sartuner first isolated morphine from the opium. You have seen in previous slide 1804. The classification of alkaloids, more recent classification are based on similarity of the carbon skeleton. For example, indole, isoquinoline, and pyridine-like structure, or biogenic precursor, ornithine, lysine, tyrosine, tryptophan, etc. Because alkaloids are biosynthesized from the amino acids. So based on the biogenic precursor, alkaloid also can be classified. There is no unified rule. However, they require compromises in borderline cases. For example, nicotine contains a pyridine fragment from nicotinamide and pyrrolidine part from ornithine, and therefore can be assigned to both classes. So some of the compounds are difficult to place in the single class. So alkaloids are often divided into the following five major groups. True alkaloids. True alkaloids contain nitrogen in the heterocycle and originate from amino acids. Their characteristic examples are atropine, nicotine, and morphine. Nicotine is very famous, present in tobacco. This group also include piperidine alkaloids, conine, and coniceine, although they do not originate from the amino acid. Protoalkaloids contain nitrogen and also originate from amino acids. Example include mescaline, adrenaline, and ephedrine. Polyamine alkaloids, derivatives of putrescine and spermidine and spermine. These are, putrescine, spermidine, and spermine. They are very important in plant as antioxidant, and they help plant to defend against different stresses. So ecologically, they are very important. Peptide and cyclopeptide alkaloids. There are lots of peptide and cyclopeptide alkaloids. If you browse in the Google, you can find lots of peptides and cyclopeptide alkaloids. Pseudoalkaloids, alkaloid-like compounds which do not originate from amino acids. This group includes terpene-like alkaloid or steroid-like alkaloids, as well as purine-like alkaloids, such as caffeine. Caffeine, you know very well, in the coffee. Caffeine is the main active, natural product, neurostimulating compound, and caffeine has also antioxidant principle. This is why coffee is considered the best drink in the world. There were lots of literature published in Nature, Science, and lots of work have been done. Theobromine and theophylline. And you must understand that although coffee is the best uh, drink in the world, but you need to be careful about the dose, how many times you are taking the coffee. Too much coffee, too much anything is bad. So this is the last slide. Tropane alkaloids of the nightshades. Tropane alkaloids, nightshade, deadly nightshade, Atropa belladonna, um, available in our country also, and other plants of Solanaceae. They produce racemic mixture of the hyoscyamine and L-hyoscyamine. And these are some atropine, and they are medicinally very important. Competitive agonist of muscarinic acetylcholine receptor. So neurologically, they are very important compound. Blocks contraction of circular pupillary sphincter muscle controlled by acetylcholine release, used as mydriatic to dilate the pupils. It can help the concentrate something. Also used by troops as an antidote for poisoning by nerve gases that target acetylcholine receptor. Used by Egyptian Queen Cleopatra to dilate her pupils in the hope that she would appear more alluring. Belladonna is the Italian word beautiful lady. So Atropa belladonna produce this type of compound, atropine. They are neurologically a very powerful neurostimulator or neuroregulator, and these compounds have huge medical application. And since ancient time, even Queen Cleopatra used this type of compound. Another compound is scopolamine, obtained from henbane, Datura stramonium, Datura plant in Bangladesh. Datura plant contain lots of alkaloids, and some of alkaloids have been used by thief or robber to make people unconscious. Other plants of the Solanaceae as atropine, competitive agonist of the acetylcholine receptor. However, the therapeutic ranges is narrower and there are more side effects and very limited use. In case of Datura scopolamine, they have some side effects. This is why medicinal use is limited. So this is all for today. We can discuss more about the alkaloid in the later class. Now you can ask some question. Just unmute yourself and ask any question, even on my lecture as well as about your review. Any type of question you have, you can ask.
Sir, uh, in during what should I say, during finding papers or searching papers, we just don't get access to some interesting papers. So what can I do then?
Do you know our students also SciHub dot se? There are some mechanism to get the paper. Rahat, do you know it? SciHub.c?
Yes, sir.
So share with Fatima. So there is a website developed by Russian scientists which can breach any copyrighted paper. So what you need to do, the DUI number starts from 10 to the later. So just copy the DUI number of that paper and paste in that page like search engine. And if you enter, you can get the PDF.
স্যার সাইহাবের
Okay, sir.
হচ্ছে একটা ক্রোম এক্সটেনশনও আছে, তো ক্রোম এক্সটেনশন দিয়ে করলে আর স্যার হচ্ছে ওই আলাদাভাবে ওয়েবসাইটে যাওয়া লাগে না, জাস্ট গুগল স্কলার থেকে রাইট ক্লিক ইন ওপেন ট্যাব দিলে অটোমেটিক হচ্ছে সাইহাবে ওপেন হয়ে যায়।
Excellent. So what you do when you have a meeting, then share all this,
ইয়েস স্যার, শিওর স্যার।
tricky method, because in Bangladesh, unfortunately, we have no access of many journals, and in
Yes, sir.
scientific world, we are working for making all the journal available. That means I'm a worker and you should be a worker on open science and open data sharing. In case of coronavirus, you have seen they practiced open data sharing and open science practices. This is why less than a year we have the vaccine. And very quickly, people developed real-time PCR and other methods, because all the papers published on coronavirus, all the sequences done by the scientists all over the world, they are open access. And this type of open access actually we practiced for the first time in science for wheat blast. So wheat blast, we have a open wheat blast website. So when you have time, please visit that one. We are not sure people, motivated by us, but we were the pioneer, and we have a story published in PLOS Biology as perspective article in case of plant health emergency. How should we address and engage the global scientific community? And similar model uh, uh, has been used for addressing the novel coronavirus causing a COVID-19 pandemic. So we are very happy to see that, and it is a new trend, so that a scientific discovery immediately can be shared by others. Do you know preprint? There are some preprint portal. So when you discover something and write a paper, immediately within 24 hours you can publish it in the preprint portal. There are for example bioarchive or other preprint portal, and the preprint portal will give you DUI number, and anybody from anywhere can get the access of the paper. But to get the credit of the publication, sometimes peer reviewed journal is very important. Preprint is not peer reviewed. This is why nowadays what we do, we publish immediately preprint and then submit to the journal. And journal initially they restricted this practice because they thought it is already published. So what is the meaning to publish it again? But now all the journals, they welcome to publish your paper in preprint and then submit to the journal. There are several advantages. One big advantage is that immediately you can share the discovery, and second, so that other people can do the research. Second is, if there is any mistake in your paper, if there is any weakness in your paper, people can make freely from anywhere. Anyone can make a comment, and even people can do a peer review. And these comments are also considered by the journal editor when accept the paper. And for the author, during review, if you get the comments in the preprint portal, then you can improve your paper and revise your paper because people are freely doing this job. So this is really a fantastic world we are now creating, open data sharing and open science. Any question?
Sir, I have a question not related to this class, but related to lab works. So why do we use gel electrophoresis instead of DNA quantification to know if DNA is present in a sample? Because gel electrophoresis is much riskier, and quantification is much easy process. So why do we use gel electrophoresis?
Yeah, in case of quantification, we use, spectrometric method, but not sure exactly the DNA or the other molecules present there. So you can do the quantification, and we use a nano drop or other device. But in case of gel electrophoresis, small amount of compound, if you use a nucleic acid, you can clearly see the molecular weight, and you can be confirmed that it is the your target DNA. This is the advantage, and it is a classical technique we have been doing, and very clear evidence. But in case of quantification, um, the purity you cannot understand. But in gel electrophoresis, if you see the band, if DNA is degraded, for example, partially degraded, you can find the ladder of instead of a clear cut band or like that. So gel electrophoresis has some advantages, and it has been used since long, considering its advantages. But we need to go.
Another question is how much DNA is sufficient in a sample for whole genome sequencing?
It depends. It depends on the size of the genome. If genome size is bigger, you need a higher amount for deep sequencing. And the sequencing, in case of whole genome sequencing, or if you want to sequence a particular fragment, in that case, after PCR, you can do a PCR product, you can use even small amount is no matter. But in case of whole genome sequencing, the higher amount, uh, uh, you know, several 10 nanograms is better. Otherwise, you cannot get uh, good sequence data. Good question.
Thank you.
And when you do the sequencing in our lab, try to learn the whole steps, and also the people who are coming from the Illumina, they know very well about the sensitivity of their machine. It depends on the machine also. So you can
Yes,
ask
sir.
more
We
questions.
are recording videos of their
Yes,
work and
excellent. And
trying
you
to
can,
take notes from them.
yes, excellent. And notes, what you do, Rahat, please type the notes, not only in the notebook, the steps
Yes.
of DNA sequence by Illumina, NextSec 550 or MiniSec. Please make, a electronic copy with your video. That would be fantastic.
Yes, sir. I will do that next time.
Okay. Any
Sir,
more?
I have another class at this time.
Yes, sure. I am going to stop my class now. So, any more question? If no question, then thank you so much. And if you have another class or another appointment, please do the job and have a good day. And I have a class with Shafi at 11:30, right? Shafi, are you there?
Yes, sir. Maybe 11:45.
11:45. Okay, fine. And then 11:45, I can take a short break. Thank you so much. Have a good day.